Welcome back to the University of Melbourne Biology series. This is episode 7, and it tells the story of two conversations. The first is the conversation between a leaf and the sun, the process by which light becomes living matter. The second is the conversation between cells themselves, the molecular messages that let one cell tell another what to do. At first these sound like separate topics, yet both are really about the same thing: capturing information or energy from outside, converting it into a usable form, and producing a response. Over the next 12 minutes we will follow energy from sunlight all the way into sugar, see how plants adapt that process to harsh environments, and then turn to how cells talk to one another. Let us begin where almost all life on Earth begins, with light. Start with the overview. Photosynthesis is a complex process that runs in two main stages. The first stage is the light-dependent reactions, which convert light energy into chemical energy. The second stage is the Calvin cycle, which uses that chemical energy to convert carbon dioxide into glucose. Hold on to that division, because it is the key to the whole episode. One stage captures energy from light, and the other spends that energy building sugar. The payoff is enormous. This single process is crucial for the production of oxygen and organic compounds across the biosphere. Almost every meal you have ever eaten, and much of the oxygen you have ever breathed, traces back to these two linked stages running inside a leaf. Before we follow the energy, let us pin down where all of this takes place. Photosynthesis occurs in the chloroplasts, and not just in plants. It happens in the chloroplasts of plant cells, in algae, and in cyanobacteria. Within a leaf, chloroplasts are not spread evenly. They are most abundant in the mesophyll cells, the inner tissue of the leaf. And inside each chloroplast sits the molecule that makes the whole process possible: chlorophyll, the primary pigment that captures light energy. So the address of photosynthesis is precise. Light energy is captured by chlorophyll, packed into chloroplasts, concentrated in the mesophyll cells of leaves. With the location fixed, we can now watch what that captured light actually does. Now to the first stage, the light-dependent reactions. Their job is to convert light energy into chemical energy, in the form of two carrier molecules, ATP and NADPH. How? It begins when light energy is absorbed by chlorophyll. That captured energy then drives a remarkable step: the splitting of water molecules. As water is split and the energy is processed, the reactions generate ATP and NADPH, the chemical energy carriers that the next stage will need. Notice the pattern from our opening theme. Energy arrives in one form, as light, and the cell converts it into another form it can actually use, as chemical bonds in ATP and NADPH. The sun's energy is now packaged and portable, ready to be spent. Let us put the light reactions on a simple ledger of what goes in and what comes out. On the input side there are three things: light energy, water, and carbon dioxide. On the output side there are three things as well: oxygen, ATP, and NADPH. The oxygen is released as water is split, and it is the very oxygen that fills our atmosphere. But the two outputs that matter most for the plant are ATP and NADPH, because those products are then handed on to the Calvin cycle. This is the crucial link in the chain. The light reactions do not build sugar themselves, they generate the energy currency, ATP and NADPH, that the second stage will use to do exactly that. Let us follow that currency into the next stage. The second stage is the light-independent reactions, better known as the Calvin cycle. The name is a useful warning: these reactions do not require light directly. Instead they run on the ATP and NADPH produced by the light reactions. The Calvin cycle is a series of enzyme-mediated reactions that occur in the stroma of the chloroplast, the fluid that surrounds the inner membranes. Its overall purpose is to convert carbon dioxide into glucose. The cycle works in steps. Carbon dioxide is fixed onto a 5-carbon sugar called RuBP, and the resulting 3-phosphoglycerate, or 3-PG, is then reduced to a product called G3P. So the light reactions deliver the energy, and here that energy is spent turning inorganic carbon dioxide into the carbon backbone of sugar. Let us slow down on the single most important step in that cycle, carbon fixation. Carbon fixation is the process by which atmospheric carbon dioxide is converted into organic molecules. It is the moment inorganic carbon becomes part of life. The key player is RuBP, a 5-carbon sugar that acts as the primary acceptor of carbon dioxide. When carbon dioxide is fixed onto RuBP, it forms 3-phosphoglycerate, the 3-PG we just met. Why does this one step matter so much? Because the fixation of carbon dioxide is the first step in converting inorganic carbon to organic molecules, and that process is essential for the production of glucose and other carbohydrates. Every carbohydrate in a plant, and ultimately the food chains that depend on plants, begins with carbon dioxide latching onto RuBP. Carbon fixation is not flawless. Sometimes the cycle misfires, and the result is photorespiration. Photorespiration happens when RuBP is incorrectly fixed by oxygen instead of carbon dioxide, producing a molecule called phosphoglycolate. This is an inefficient process, and it can reduce plant productivity. So when does the mistake happen? Photorespiration is more likely under conditions of high oxygen and low carbon dioxide, such as on hot, dry days. And it is more common in certain plants, called C3 plants. Think about what a hot, dry day does to a leaf. It must limit water loss, but doing so chokes off its carbon dioxide supply while oxygen builds up inside. That is exactly the high oxygen, low carbon dioxide setting that triggers wasteful photorespiration. That problem sets up the next part of our story. Plants have evolved different photosynthetic pathways to optimize carbon fixation under different environmental conditions, and there are 3 to know. C3 plants use the standard Calvin cycle. They are efficient in moderate climates, but, as we saw, they are the ones most prone to photorespiration. C4 plants use a modified Calvin cycle, with an initial carbon dioxide fixation step in the mesophyll cells. This gives them a more efficient carbon dioxide fixation process, adapting them to hot and dry environments. CAM plants take a different route again. They fix carbon dioxide at night and store it as malate during the day. By opening their stomata at night, CAM plants conserve water, which is why they are found in arid regions. Same Calvin cycle at the core, 3 elegant solutions to 3 different climates. Now to the episode's second conversation, the one between cells. Cell signaling is the process by which cells communicate with each other. It involves detecting chemical signals and transducing those signals into cellular responses. Cells use several types of signal, depending on how far the message must travel: autocrine, juxtacrine, paracrine, and hormonal signals. But here is the crucial idea. Specificity in signaling is achieved through the interaction of ligands with specific receptors, and different cells respond to the same signal differently because of variations in receptor expression and in their intracellular pathways. There are also 2 broad places a signal can be detected. Membrane receptors handle large or polar ligands that cannot cross the membrane, while intracellular receptors handle small or nonpolar ligands that can. The chemistry of the messenger decides the route. Let us zoom in on the receptors that sit at the cell surface. These are proteins that bind a specific ligand and then initiate signal transduction. They come in 3 main classes, each with its own mechanism. The first are ion channel receptors. When their ligand binds, they allow the passage of ions across the cell membrane. The second are enzyme-coupled receptors. When their ligand binds, they activate enzymes, setting off a cascade of intracellular events. And the third are G-protein-coupled receptors. When their ligand binds, they activate G-proteins, which in turn modulate intracellular signaling pathways. 3 different switches, but the same logic in every case. A signal arrives on the outside, the receptor changes, and a message is passed to the inside of the cell. Once a receptor passes the message inward, what happens next? The signal enters a transduction pathway, a complex network of interactions that relays signals from the cell surface toward the nucleus. These pathways do far more than simply carry a message along. They can relay the signal onward, and they can amplify it, so that a small input produces a large response. They can integrate multiple signals, weighing several inputs at once. And they can distribute the signal to multiple effector proteins, fanning one message out into many actions. That versatility is the point. These pathways are essential for coordinating cellular responses to external signals. So our two conversations end up sharing one deep theme. Whether a leaf is turning sunlight into sugar, or a cell is turning a chemical signal into action, life works by capturing information, converting it, and producing a coordinated response. See you next time.